Hello, everyone, and welcome to today's webinar titled Identification of Human Immune Cell Subtypes Most Vulnerable to IL-1β-induced Inflammatory Signaling Using Mass Cytometry. I'm Cindy Valdez of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is sponsored by Fluidigm. So let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you want during this presentation. To do so, simply type them into the Ask a Question box and click Send. We will answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing this presentation, just click on that Ask a Question box located on the far left of your screen. I now present today's speaker, Dr. Hema Kothari, an assistant professor at the Department of Medicine, Cardiovascular Medicine Division at the University of Virginia. For a complete biography on Dr. Kothari, please visit the Biography tab at the top of your screen. Dr. Kothari, you may now begin your presentation. Welcome.
Thank you for the introduction, and hello everyone out there watching this presentation. In today's talk, I'll be sharing data on how we utilize the powerful mass cytometry approach to identify the immune cell subtypes in humans that are targeted by the pro-inflammatory cytokine IL-1β. We hope that this work and other ongoing projects in our lab will contribute towards a better understanding of the inflammatory immune response to COVID-19 and help develop potential strategies for assessing disease severity and response to therapy for better outcomes. Before I show you our data, I'll talk briefly about the cytokine storm in COVID-19, the role of IL-1β in cytokine storm and tissue injury, and then I'll discuss few recent findings that show the potential of targeting IL-1β for treating the cytokine storm and mortality. The COVID-19 cases across a number of countries are still on the rise. Here is a snapshot of the number of the confirmed COVID-19 cases between June 17th to June 23rd across different countries. And if you go back and check on the WHO site, you would realize that just in this past week, there has been an increase of 10% in the number of confirmed cases and about 6% increase in the number of deaths. COVID-19 infected subjects show a range of clinical symptoms. Some of them, while infected, remain asymptomatic, while others develop mild to moderate disease and recover. And then there are a subset of individuals that have severe outcomes such as pneumonia, ARDS, multi-organ failure, and death. We know that people with comorbidities such as hypertension, hyperlipidemia, diabetes, and other underlying conditions, and the elderly are at higher risk. Recent studies suggest that immune dysregulation and cytokine storm lead to disease severity and mortality in COVID-19. Shown here in the figure is the immune response in subjects with mild to moderate disease. So the way the SARS-CoV-2 virus gains entry into the cell is through binding to the ACE2 receptor, which is expressed on lung epithelial cells, and then through cleavage of its spike protein by the serine protease TMPRSS2. In addition, pattern recognition receptors on innate immune cells, such as macrophages, can also sense the RNA viruses. In most individuals, the interaction of this virus triggers an efficient immune response, such as production of type 1 interferons, shown over here in yellow, and other pro-inflammatory cytokines such as TNF-alpha, IL-1β, IL-6, and others. This helps clear the viral infection with minimal inflammation and tissue damage. However, in high-risk subjects with severe disease, the antiviral response is not efficient enough and leads to an uncontrolled viral replication and a dysfunctional immune response. This in turn drives a massive production of cytokines that leads to severe lung inflammation, coagulopathies, organ failure, and death. IL-1β is one of the inflammatory cytokines, higher levels of which are present in COVID-19 patients with severe disease. The different mechanisms through which IL-1β can contribute to severe disease is through causing cell pyroptosis, production of other downstream inflammatory cytokines such as IL-6, TNF-alpha, interferon gamma, and so on, and induction of the coagulation protein tissue factor on macrophages, lung epithelial cells, and endothelial cells. IL-1β can also recruit immune inflammatory cells to the site of inflammation and can induce immune cell proliferation and differentiation. In addition to IL-1β, there are other cytokines and chemokines. Some of them that I have listed over here are being shown to be associated with severe disease and mortality in COVID-19 patients. The way IL-1β influences the cellular functions is through binding to its cell surface receptor, IL-1 receptor 1, and its interaction with the co-receptor, IL-1 receptor 3. This trimeric complex then triggers a series of intracellular events that ultimately leads to the activation of NF-kappa B, AP1, as well as MAP kinases and drives the gene transcription. There are different inhibitors of IL-1 that are used in the clinics, mostly for treating chronic inflammatory diseases such as rheumatoid arthritis, gout, and other autoinflammatory diseases. One of them is Anakinra, which is an IL-1 receptor antagonist, and the way it blocks IL-1β signaling is by binding to the cell surface receptor IL-1 receptor 1. I want to mention that Anakinra can inhibit signaling mediated by both forms of IL-1, which are IL-1 alpha and IL-1β. Canakinumab, on the other hand, is a monoclonal antibody that is specific to IL-1β. A third inhibitor of IL-1β used is rilonacept, which is basically a fusion protein, and similar to Anakinra, it can also inhibit signaling mediated by both isoforms of IL-1. Given the evidence of the cytokine storm in severe COVID-19 infection, multiple clinical trials are testing the potential of anti-inflammatory agents such as inhibitors of IL-1β, IL-6, and JAK signaling proteins currently. Two of the recent studies that were published show the potential of Anakinra for reducing inflammation and mortality in COVID-19 patients. One of them is the study by Thomas Hugh et al. From France, which assessed the off-label use of Anakinra in patients with severe forms of COVID-19. In this study, they used two cohorts, a prospective cohort of 52 subjects that were treated with Anakinra 100 milligrams twice a day for 72 hours, and then 100 milligrams daily for 7 days. The control cohort, enrolled retrospectively following the same inclusion criteria, was treated with the standard therapy alone. The results showed significantly lower risk for death or admission to the ICU for invasive mechanical ventilation in patients treated with Anakinra as compared to the control group. A second study by Cavalli G et al. From Italy also showed positive outcomes with Anakinra. The three arms in this trial were a cohort of 16 patients that just received the standard therapy, a cohort of 7 patients that were treated with 100 milligrams Anakinra twice a day for 21 days, and then a third cohort of 29 patients that received a high dose of Anakinra for 21 days. In this study, lower dose Anakinra treatment was discontinued after 7 days because there was no effect on serum CRP levels and clinical status. However, higher dose Anakinra treatment showed clinical improvement in terms of increased survival and reduction in the need for mechanical ventilation. The possible reason for the discrepancy in the outcomes with low-dose Anakinra between these two studies could be the differences in the comorbidity status of the control group. As in the other study by Thomas Hugh et al., the authors did mention that their control group comprised of subjects that were more obese, and that could possibly have confounded the results. While IL-1β inhibitors are emerging as a potential therapy for inflammatory disorders, it is unclear which specific cell types in humans IL-1β targets and whether it activates different signaling pathways in different immune cell subsets. To answer these questions, we designed a custom 35 antibody CyTOF panel consisting of cell surface markers for broad identification of major immune cell subsets, IL-1 receptors, and intracellular phosphosignaling proteins. We then performed optimizations. Our future goal is to use this panel for immune phenotyping PBMCs from subjects in clinical trials. Therefore, we first confirmed expression of markers in cryopreserved PBMCs by comparing them with fresh, and results were very comparable. All antibodies were titrated, and we ensured that there is no bleed-through that may arise as a result of metal impurity or metal oxidation. Finally, as we were interested in detecting transient phosphorylation events and the cells needed to be fixed, we evaluated the effect of PFA fixative as well as methanol that is used for cell permeabilization on cell surface marker epitopes. We found that PFA alters antibody reactivity, and this was especially the case for the chemokine receptors as shown over here. The staining was either severely diminished, such as for the case for CCR7, CXCR5, or enhanced non-specifically, as you can see here for the chemokine receptor 3, CXCR3. In order to stain PBMC samples with these PFA-labile antibodies, we then adopted an alternative strategy where we added these antibodies at the same time of cell stimulation with IL-1β and incubated the cells with the antibodies for 15 minutes. We evaluated if antibody addition affects the signaling outcome and found no effect, as you can see over here in these two figures, where left was with no antibodies added, and in the B figure, the antibodies were added at the same time of IL-1β stimulation, and there was no change in the frequency of the phospho-NF-kappa B positive cells. Here is our experimental setup that we used for our studies. We used cryopreserved PBMCs from healthy donors that were thawed, rested, and stimulated with IL-1β in the presence of the PFA-labile antibodies. Cells were then fixed with PFA and barcoded before stained with the cell surface or other intracellular antibodies. Samples were run in CyTOF, and the acquired data was then analyzed using the R-based computational algorithms, cytobank, and by traditional gating. To identify the immune cell subtypes that respond to IL-1β stimulation, we first performed SPADE analysis in cytobank. SPADE is an algorithm to analyze multi-parameter data, and the way it works is it basically puts the phenotypically similar cells in a cluster and then generates a 2D tree of those clusters. Figure A, shown over here, shows the localization of the different immune cell subsets, such as CD4 T cells, CD19 T cells, CD8 T cells, and so on. The size of each circle and color in this particular figure depicts the number of the cells. So blue color depicts less cell count, and red color, shown over here, depicts higher cell count. The clusters were defined as CD4 T cells, CD8 T cells, B cells or monocytes, and so on, based on the median expression of the lineage markers as shown in figure B. The color here, blue to red, represents low to high marker expression. Figure C shows expression of phospho-NF-kappa B, phospho-P38, and phospho-ERK across all immune cell subtypes in vehicle-treated, which is the top panel, and the IL-1β stimulated samples, which is the bottom panel. Here, the treatment was, this figure is coming from the cell samples that were treated with IL-1β for 15 minutes. As you can see, IL-1β induced NF-kappa B activation within the CD4 T cells, CD4 negative, CD8 low T cells, CD11C myeloid dendritic cells, and to some extent in the natural killer cells. P38 activation was observed primarily in the CD14 monocytes, CD11C dendritic cells, and to some lower level in some of the CD4 T cells, as seen here within the CD4 bubble. As compared to vehicle, there was no change in the ERK activation upon IL-1β stimulation. I just want to emphasize one more thing, that the scale over here for the 3 different phosphoproteins are different. So although the color red is there, but the level of ERK activation, either basal or under stimulated conditions, was lower as compared to phospho-P38 and phospho-NF-kappa B. To identify the specific immune cell subsets, even within what are the specific cell subsets within CD4 T cells or within the monocytes, we then performed UMAP and Leiden clustering. We clustered the T cells, monocytes, and NK cells separately, and the cells that we used for our clustering analysis are shown in the figure. So we used this particular cells gated with this gate as shown in figure D for T cells, and then as shown in figure E, these 2 gates, we used either the CD56 positive HLA-DR negative cells for the natural killer cell clustering or the DR positive cells for monocyte and dendritic cell clustering. We identified 12 distinct T cell clusters, and when we projected the clusters on a UMAP, which is a model of dimensionality reduction that condenses everything down to 2 dimensions, you can see that the CD4 T cells shown here on top of the UMAP and the CD8 T cells, the clusters 2, 10, and 11 at the bottom of the UMAP, they were distinctly separated from each other as anticipated. We then determined the frequency of these clusters in the subjects, which is shown in figure B, and as anticipated, there was heterogeneity in cluster abundance between the subjects. Phenotype of the T cell clusters was annotated based on the expression of the markers as shown on the heat map here in figure C, where blue to high color in the scale depicts low to high expression. And in addition to the heat map, we also used the violin plots to annotate these clusters. The violin plots are not shown on the slide. As an example, we identified cluster 12 as memory Tregs based on the expression of CD4, CD25, CD45RO, and the lack of expression of CD127. Having annotated the clusters, we identified robust activation of NF-kappa B in clusters 3, 4, 8, and 12 in response to IL-1β. These clusters corresponded to Th17 cells, T follicular helper cells, CD4 negative, CD8 low, CD161 high T cells, and memory Tregs. We also found activation of P38 within the same clusters, but the level of activation of P38 was much lower as compared to the NF-kappa B. Interestingly, we identified that all of the 4 subsets that respond to IL-1β are effector memory T cells as defined by the expression of CD45RO and lack of expression of CCR7. Next, we were curious to see if the expression of IL-1 receptor 1, which transmits IL-1β signaling, is limited to the cells that respond to IL-1β. To test this, we separated total T cells into phospho-NF-kappa B negative and phospho-NF-kappa B positive populations and then measured expression of IL-1 receptor 1. Surprisingly, we observed a significantly lower expression of receptor 1 on the phospho-NF-kappa B positive cells as compared to the phospho-NF-kappa B negative cells in cells that were treated with either vehicle control or IL-1β. This data suggests a difference in the expression of other proteins, either cell surface or intracellular, that may regulate IL-1β signaling response. IL-1 receptor 2 is another cell surface protein that binds IL-1β, but it acts as a decoy receptor in that it doesn't transmit the downstream signaling. So thus, the ratio of receptor 1 and receptor 2 on a cell may affect the final signaling outcome. Unfortunately, we could not detect IL-1 receptor 2 in our CyTOF analysis. However, using the same antibody clone, we were able to measure IL-1 receptor 2 expression in PBMCs that were cultured in the presence of anti-CD3, anti-CD28, and IL-2. And you can see over here in the figure on the right-hand side, the middle panel, that even some level of IL-1 receptor 2 expression was seen in cells that were cultured for 48 hours but were not stimulated with the T-cell activating factors. Shown here on the rightmost is the FMO control that we use to gauge the IL-1 receptor positive population. A deeper look to identify the unique marker that marks the T cells that respond to IL-1β, we found that expression of CCR6 is a common feature among the 4 clusters that showed signaling activation. Although the expression of CCR6 varied between the 4 clusters. To find out if CCR6 expression would identify the responsive cells, we separated the total T cells into CCR6 negative and CCR6 positive T cells, and then analyzed for the expression of phospho-NF-kappa B and phospho-P38. Interestingly, as shown in figure B over here, the NF-kappa B positive cells were enriched within the CCR6 positive T cells. We observed the same with phospho-P38 positive T cells. Furthermore, up to 60% of the CCR6 positive CCR6 positive T cells that responded to IL-1β stimulation also co-expressed CCR2 and CD26, as shown here in the UMAP overlays. Following similar steps, we identified the monocyte clusters, annotated them, and determined their abundance in different subjects. We identified NF-kappa B activation in the CD14 classical monocyte subsets, cluster 1, 2, 6, and 9, as well as in the CD11C myeloid dendritic cell, which is cluster 8, shown over here. In addition, IL-1β also stimulated NF-kappa B, phospho-NF-kappa B in cluster 12, which was found to be a lineage negative cell that only expressed CD38. We don't know what that cell type is yet. P38 activation was also observed within these same subsets. For clustering NK cells, we use the classic broad NK cell markers such as CD56, CD16, and CD161, and also included other markers as shown over here on figure C heat map that we found that were expressed on the NK cells. After clustering, we identified 17 clusters and found NF-kappa B activation induced by IL-1β in clusters 2, 3, and 17. Interestingly, cluster 17 that showed the most robust activation of NF-kappa B and P38 was found to be the one that uniquely expressed CD25. Cluster 2 corresponded to the CD56 dim, CD16 dim population, and cluster 3 was the CD56 bright, CD16 negative population. This cluster 17 that expresses CD25, we suspect that these cells could be either some sort of innate lymphoid cells or activated NK cells, but further studies are needed to confirm their identity. We also evaluated indirect signaling mediated by IL-1β by stimulating cells for longer time points. As you can see, IL-1β stimulation led to a transient early activation of NF-kappa B in T cell subsets shown over here that peaked at 15 minutes and then was reduced to basal levels at later time points. Monocytes, CD11C dendritic cells, and NK cell subsets also showed similar kinetics of NF-kappa B and P38 activation, but it's not shown over here. In contrast, longer stimulation with IL-1β for 1 hour or more led to differential activation of STAT1, STAT3, and STAT5 in subsets of T cells, monocytes, and NK cells. Shown here is the data for STAT3 activation in T cell subsets, which clearly shows the donor heterogeneity in the response, as well as the differential activation in the different T cell subtypes. For detailed information for people who might be interested in knowing more about the specific immune cell subsets and the direct and indirect signaling response mediated by IL-1β, please refer to our online manuscript in the bioarchive. So finally, as anticipated, we found that Anakinra inhibits IL-1β-induced signaling, as shown here for NF-kappa B activation in the CCR6 positive T cells, CD14 positive monocytes, CD11C myeloid dendritic cells, and the CD25 expressing NK cell cluster. We have other ongoing projects in the lab that are focused on testing and developing immune-based signatures for personalized anti-inflammatory therapy for CVD. And I thought that I'll just share this small piece of data that was done in the heart failure subjects. So similar to the healthy subjects, in the heart failure subjects, IL-1β induced NF-kappa B activation, specifically in the Th17 cells, as well as in the CD11C myeloid dendritic cells. The interesting finding here is that there is remarkable heterogeneity in response to IL-1β stimulation, as well as to inhibition by Anakinra. We anticipate seeing such heterogeneity in the COVID-19 infected patients, and we believe that immune phenotyping these subjects using a similar approach may discover immune biomarkers for early identification of COVID-19 patients at risk of cytokine storm and those that might respond to agents that block cytokine storm. So finally, to summarize, we have identified the immune cell subsets that respond to IL-1β. CCR6 expression was found to be as a marker for identifying the T cells that respond to IL-1β. IL-1β induced NF-kappa B activation more prominently than P38 in T cells, dendritic cells, and NK cells. We did not find activation of ERK or JNK by IL-1β stimulation in any cell subtype. And as compared to other immune cell subsets, B cells seems to be quite unresponsive to IL-1β stimulation. And we think that the activation of STATs that we observed at later time points after IL-1β stimulation is most likely arising as a result of the production of other downstream pro-inflammatory cytokines such as IL-6, interferon gamma, or GM-CSF. And that work is currently ongoing in the lab to look more into it. Finally, for my acknowledgements, I'm really thankful to my mentor, Dr. Colleen McNamara, as well as all the wonderful people in her lab that makes work really fun and exciting. For the IL-1β specific work, Chantal McScheming and Maithili Vigneshwar really contributed significantly for that project. I'm really thankful to my collaborator, Dr. Eli Zunder in the Biomedical Engineering Department, and his PhD student, Corey Williams, for their help with data analysis. And finally, I would like to acknowledge the wonderful flow cytometry core people at UVA and AHA for supporting this work. Thank you.
And thank you, Dr. Kothari, for that informative presentation. And we will now start the Q&A portion of the webinar. Audience members, if you have questions you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. So it looks like, Dr. Kothari, that we already have some questions coming in from our audience members. Our first question, what do you think are the reasons for the variability in the donor response to IL-1β?
Yeah, that's a great question. And as we initially we thought that it could be the level of the receptor 1 or the receptor 2 present on the cell surface that will determine the response in the donor. But then, like, based on our T cell data, it kind of like suggests that it is a little bit independent of the receptor 1. And there could be multiple factors for that. One of them is we think that definitely there will be it is genetically encoded. So we know that there is polymorphism in IL-1 receptor as well as the other intracellular signaling proteins such as MYD88. And so that could be one of the really possible reasons that why subjects show differential response. And then what we have not currently measured is the level of the other signaling proteins as well as the co-receptors that mediate the signaling. So in all, I think the interaction between them dictate the signaling outcome. So the levels of the different receptors and the co-receptors, I think also makes a difference in the signaling outcome, and that's how that impacts the donor heterogeneity in response to IL-1β.
Thank you for that response. One audience member wants to know, do you plan to also measure downstream intracellular signaling proteins involved in the IL-1β pathway?
Yeah, we do, and we have spent some time now trying to do that one, and one of the major problem is that we don't have the antibodies, like good antibodies that are compatible for flow or site-off. And we have kind of like tried to find different antibodies. We have some that we are currently validating, but it is tough. I just hope that the Fluidigm people or other company people like come up with these other antibodies that are really important, such as for the IL-1β pathway or IL-6 pathway, because that would make our work really easier, and it would provide more insights, deeper insights into what's exactly going on. So yeah, we are looking forward to it, but some work is ongoing, and we don't have all those antibodies.
Thank you, Dr. Kothari. And again, thank you to our audience members. Any questions not answered today will be answered via email. We have time for a couple more questions before we close. How did you decide to use UMAP as opposed to all the other clustering algorithms available?
Yeah, so that's a good question. So we use SPADE, and I personally like to use SPADE because when we are working with, example, looking at intracellular signaling, and our question is like how it impacts the different cell subsets. So SPADE provides a quick overview. It is to look at what's going on across different immune cell subsets. And then we can go on into more deeper other clustering algorithms, which are more complicated, but at the same time, they provide more detailed information, and it is also much easier. I know, I mean, with SPADE, you can still get the more quantitative data from each node and other things, but it is, I think, to me, it is a little bit more time-taking as compared to using other clustering algorithms like Leiden or Louvain, if you have the right people to help you out with those analysis.
Thank you so much. One last question. Is anyone studying the IL-1β pathways in COVID patients with adverse heart impact?
So my answer to that question would be, I think it is very much likely. I'm not completely aware of that one, but we know that a lot of the patients who are getting severe symptoms after COVID-19 infection, they have these comorbidities, and a lot of them have cardiac issues. So I think that's a valid reason, and inflammation is a common pathway for the pathogenesis of both. And we are seeing that in COVID-19 infected patients, a lot of them are dying due to these coagulopathies or heart-related problems. So that's an important area to study, and I believe like somebody is definitely studying that.
Dr. Kothari, thank you again for today's presentation and clearly important research. I also want to thank Fluidigm for sponsoring today's event and encourage audience members to check out other presentations and explore the virtual environments and booth. And before we go, I want to thank audience members for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand through July of 2021. Until next time, have a great day. Stay safe. Bye-bye.
Thank you. Thank you.
Thank you.